Using open source tissue image analysis tools for life science and biomedical research is a no-brainer. There are already tens of thousands or probably even hundreds of thousands of scientists using those tools. Using those tools for commercial purposes is a different story, and building a platform for tissue image analysis based on those tools is something I have only recently heard about from my today's guest. When I was investigating open source software for analyzing pathology images, this premise of open source being available for commercial purposes sounded super attractive, but I didn't really know how it can be used. And my guest is going to talk about it today. So let's dive into it.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Welcome, Digital Pathology Trailblazers. Today, my guest is Trevor McKee. Trevor McKee is the CEO of Pathomics. Pathomics is an image analysis company, but it's a different kind of image analysis company because they are using open source software to do image analysis. Welcome, Trevor, to the podcast. How are you today?
Thanks, Alex. Really great to see you. I'm doing super well. Thanks.
We have met. Pathomics is not your first image analysis rodeo,
Yes.
so to say, because we have met at several companies before. I'm going to let you introduce yourself and let the listeners learn about your background.
Okay, that sounds great. So I'm a renegade chemical engineer that didn't want to go into petroleum. And so I got interested in biology in undergrad. Yeah. Oh, yeah.
My husband started as a chemical engineer and worked at in some glue company, and then he became an MD. So now he's a doctor.
MD. Oh, very cool. Very cool. Yeah. So I mean, engineering in general, chemical engineering is very good sort of general purpose problem solving that you can apply to a lot of different things, right? So I really got into biology in grad school, where I went to MIT biological engineering. And there I worked in the lab of Rakesh Jain, who is a pioneer in sort of biomedical imaging and intravital microscopy. And so I did a lot of 2-photon imaging in mouse models of cancer through my work there, and really found that, the easy part is actually taking the images. The hard part is how to analyze and get the information out that you need, right? So from then on, I've been working kind of in that area. So I did a postdoc up here in Toronto, and then joined a core facility that called the STAR facility, which had sort of a micro CT, micro MRI, and then a correlative histopathology lab. So we could do all of the sort of imaging you could do in patients, but on small animals, and then do the pathology to register with those imaging modalities. So I was the director of the image analysis core within that core facility. And so for about 10 years, we had worked on really any sort of image analysis that came through the door, we would analyze for people. And then we purchased the Definium software in about 2016, I think. And so we were using that and really got to developing a lot of tools there using Definium to analyze, again, those different imaging modalities, but focusing a lot on sort of the histology and multiplex images. Because I was part of a big preclinical trial with Pfizer, where we were testing out a couple of Pfizer's drugs on patient-derived tumor xenografts that we were growing in mice. And so I ran a study where we were doing kind of PET imaging, CT imaging, and then taking the tissue out and staining it for a couple of markers for proliferation and hypoxia, and then looking at all of those relationships across all of the different tissues. And so that was interesting. Worked there for about 10 years, then moved to HistoWiz, where I was director of image analysis for about a year over COVID, and then moved to Deciphex, where I worked as sort of helping with commercialization of AI services. And then as of November, I've been running Pathomics and essentially trying to do what we were doing at STAR in terms of providing image analysis services to people, as well as sort of building our goal here at Pathomics really is to build a online platform that can serve as a tool for either data scientists or pathologists or people working in this field to be able to go all the way from images to analyzed images to reports that could say something about, the content of those images.
And you are going to be using, or you are using, the open
Open
source
source.
software
Yes.
for your services.
Yes.
Why and how are you doing this?
Well, that's a great question. So, I mean, for one, there's great open source tools out there, right? So,
Mhm.
so QuPath, for example, is one we've used quite extensively, and QuPath, with the right sorts of scripting added in, will do a lot of really can handle a lot of things. Can handle multiplex
Mhm.
data. You can do single cell segmentation. You can extract a bunch of features from those images and then use those for any of the downstream analytical processing you want. And I think we, both myself and my co-founder Mark, we really feel passionate about the fact that like open source software is the future. It's something that
Mhm.
everyone, as opposed to, let's say, a closed source solution, requires developers to go in there and figure out what they're doing. QuPath has the forums, right? The great image analysis forums. And actually,
I'm a member.
yes, and my co-founder is Mark Zaidi, and he's I think he might have even corresponded with
Yeah,
you
we
on
have
there. So
corresponded through the forum. Yes,
yeah,
yes. Before
so
I
he
even knew that you guys were working together.
yeah. No, he's great, and he just goes on there, and anyone that has questions, you know,
I'm gonna link to the forum in the show notes as well.
oh, that would be amazing. Yeah, and that's I mean, it's a great sort of cross-platform forum for Cell Profiler, Fiji, QuPath, and others, and so it's sort of a great resource for a lot of people. It's an interesting concept of like, how do you run a commercial business with open source tools?
Yes,
Um,
and this is like a super something that interests me very much, because open source, you can do business with it, but rarely
mhm.
anyone is. So I don't know any other company that is actually using your model. So let me
Yeah,
know more about the model, the value proposition and your business model with this kind of software.
with the
It's
software,
super
yes, yeah.
interesting.
Yeah. So I think, I mean, really the value proposition that we have is right now, let's say you were a pharma company, right? And you're
Mhm.
looking at a bunch of pathology data that's flowing through your organization, right? You're going to want to do something with that data. And oftentimes that requires, of course, a pathologist has to be involved there, of course, but sometimes let's say it's a biomarker
Of
study,
course.
and you need to know how many markers are there, in what proportions, et cetera, et cetera. And so the options for you are, either hire data scientists who are expensive, and maybe commercial software, which is expensive, and use that to do your work. Or the option could also be outsource to a company, let's say like Pathomics, who has the expertise to be able to know how to analyze the data, who can do that analysis, who can present you back with a report that you can use to sort of make decisions about what the next thing you are wanting to do. Does this all using standardized open source, but standardized tools that, we can write up in such a way that it's going to cover, from a regulatory perspective, cover all check all of the boxes. So, so I think that's one value proposition. And the other one is just, even if you do have commercial software and a data scientist, many of the commercial software sort of stop at spitting out a bunch of numbers at you, right? So here's every single cell with all of the markers present within that area. But that's not the end of the study, right? What we've done at STAR over the past 10 years is move from having that export to having an actual publication ready figures for your manuscript, or sort of a report that says this biomarker has gone up or down in this clinical trial, right? So I think that's really where we come in, is to build a platform that lets our users move from images, or move from an exported data file with all of the cells, to actually having graphs and understanding of what that data says at the end of the day. So our first product that we're putting together is an online-based platform that really, if you need the segmentation done, we can do the segmentation on QuPath. But even if you've say used Visiopharm or Halo, and you've got this export, you can upload that export to the platform. We will provide you with a way to to interactively generate those graphs that you're looking for, or maybe have some preset templates that you can use that will generate at the end of the day a report, a spatial report that says we see this many cells in this region and this many cells in that region, and that's indicative of whatever, PDL1 going up or down, or CD or more CD3 cytotoxic cells near the tumor. There's going to be a couple of different outputs that people might want depending on what their study is.
So you say it's a platform. Is it a cloud-based platform, I assume? Or
yes,
is it not like browser-based that you can
browser-based?
Yeah.
Yes.
Mm-hmm.
Yeah.
Whatever the behind the scenes of browser-based and cloud-based are, but something that you can access through Microsoft Edge or Chrome, right?
Yes. And I mean, there are versions of this out there already. It's called business analytics. So there's a lot of companies that are doing, a dashboard where you can analyze a lot of complex data. And so we're building a version of that, but specifically for digital pathology.
Okay, so you're building like an interactive dashboard for image analysis based on open source image analysis tools.
Well, yeah, it's a combination of open source tools and then probably some Python code in there that we're going to be using as well.
So tell me about, like, somebody would want to work with you. How would that work? What would the workflow be? And what kind of projects would you work on? Like, give me an example.
That's a very good question. So, I mean, one of our potential clients that we're speaking with, they already have an image analysis team. It's a CRO. They already have an image analysis team. The problem they're facing is that they need to double their output over the next 6 months without increasing headcount, right? And so they need tools to make their existing data scientists more efficient with what they're doing. To be honest, a lot of their data scientists' time is spent copying and pasting graphs into PowerPoint decks or into a Word doc, right? And that's not a very efficient use of their time. So if we can automate, even if we can just automate the reporting aspects of that, right? That's something that has value. And then other clients are looking for more of a whole service solution of they've got, a pharma client that needs image analysis, that needs staining and image analysis done. And they're going to handle the staining portion, but they need help with the image analysis. And so we'll subcontract the image analysis work from them. We'll deliver, results, specify what the outputs are back to them, and then that forms, another service offering that we can provide. So, and really, we're looking at it across a whole spectrum, right? Another big area that we serve is academics, right? And so for academics, maybe they don't have as much budget for, to be able to spend on things because grant money is a little harder to come by nowadays. But what they can do is academics have access to grad students. And so we're also working on, let's say a academic rate where we would provide, a virtual machine that would have access to QuPath and then train their grad students how to do the work themselves. And then, we'll just charge for the computational costs of hosting that data, and then provide them with an ability to develop whatever scripts they need to develop and really work with them on that aspect of it, and or potentially even give them something in QuPath that they can run themselves, right? So, that's on the, let's say the low cost end through to the full service end. And we try to cover kind of everything in between.
So what would people pay for? Is it the services that you guys are providing, or is it the access to the platform, or a combination, or something totally different?
It's certainly a combination. So, I think there's a phrase for startups that you're building the plane as you're flying it. So right now, we're working on a version of the platform. So right now, we're going to be providing a lot more services-based work, where, you provide us your images, we'll provide you the analysis back. But we want to move towards this online platform, both for our external clients, but also even for us internally. It'd be great to just be able to use this platform to do our own services-based work. And as we're building it for ourselves, we'll see how it works for building it for other people too. But I think down the line, we'd like to get to having this, being able to run all of the analysis in as automated a fashion as possible. But there's a lot of complexities that go into that, right? Because there's, if you ask 10 different people, you get 10 different answers as to what what analysis looks like, right? So,
Right.
so I think that's going to be the real work over the next couple of months, is starting to define what those image analysis sort of pipelines look like, right? And then building all of the various interconnected pieces to, let's say, take a segmentation and bring it over here to do post processing, or to do dimensionality reduction and clustering, or any of the various things that you want to do. I think what we're looking at is to really map out all of those potential connections in sort of a diagram, and then be able to flow from one through to the others.
Mm-hmm. And you mentioned that this can be built in a compliant way. If this was supposed to be used for can it be used in a regulated environment? What's your plan for that? Because especially when people in the regulated environment hear open source, they're already super cautious that how can you be in compliance with open source software?
Yeah.
But the thing with open source, the only thing that is not there is not like people that can support the software. If you have the capability in-house to work with different versions, or at least that's my understanding. But definitely when you say open source in a regulated environment, people are like, I don't know if we can use it. We probably cannot use it. Tell me more about this. Do you get this question as well?
Yeah, it's a very good question. And I would say we're still early on the road there, but I have done analysis for clinical trials in the past. And that was done under GCLP kind of practices. And it really came down to sort of documentation. So, in that case, I think we were using Definions at the time for that analysis, and we just had to
which was not
record
the compliant software on its own, right?
On its own, no. But what we did was we just recorded all of the steps that we were doing to do the analysis, and we made sure that we had manual checks in there, both for kind of manually correcting any tissue classifiers that were done, and also manually checking the number of brown cells that were counted within a small ROI, and just providing some accuracy metrics back as saying that, okay, we were between the automated counts were between 80 and 120% of the manual counts. And it was a research portion of the clinical trial, right? So there was no kind of decisions that were no patient decisions that were being made off of this. It was more to understand, were these immune cells going up or down? And so I think within that framework, we were able to make it work at that time. Whether or not, it's a big shifting landscape, as I understand, with AI and digital pathology and regulatory things. So I guess we will see. I think for now, we're mostly hoping to target the sort of preclinical end of the spectrum. And as we start to get more traction, we'll see which of these pipelines can be sort of made robust enough that they could fall into something that would go down a regulatory path. But for now, I think we're avoiding the question, let's say, or we're not focusing initially on solving everything, right? We can't boil the ocean. We'll start at the research side, and then as we move, we can branch into clinic.
So you mentioned AI. Do you guys incorporate AI in any of the platform? I'm asking because I know that currently, and by AI, I mean deep learning. AI is, a huge concept.
Yes.
But deep learning and, example-based, annotation-based kind of image analysis, training of models. Does QuPath have that already? I know Pete Bankhead, the author of QuPath, was working on it. I don't know where we stand. Tell me, as a super user, what do you do with AI in Pathomics, and how do you incorporate it into open source that you're using?
Yes. So we actually incorporated at a number of different points, and I would say it's machine learning broadly. Right. So it's both sort of classical machine learning as well as deep learning. They're different for different reasons. Right. And so actually one of the tools that Mark built and is up on his GitHub is called Universal Startist for QuPath. And so the reason we built that, that is a plugin that you can run within QuPath that will run the Startist deep learning tool to do cell segmentation within a bright field or a fluorescence image, right?
Just
And so
for explanation, Stardust is to detect nuclei in cells, right? This is a
for
deep
detecting
learning-based
yes.
pre-trained model to detect nuclei.
Exactly. Yeah. So it's a deep learning based pre-trained model, but it was trained on DAPI stained fluorescence images, right?
Oh, interesting.
They've got two. I think they've got a bright field one and a DAPI one.
Mm-hmm.
But the problem we had when we were trying to use Stardust within QuPath to analyze IMC data, so imaging mass cytometry, is that Stardust didn't work very well out of the box on that data. And the reason it didn't is because just the data, like mass spectrometry based imaging, it's very different from DAPI based imaging, different resolution, different kind of, it's a 32-bit intensity range. And so that was causing challenges with being able to run it. And so what Mark did was actually build normalization and adjust the resolution of the IMC data to make it look like DAPI data, so that when you run Universal Stardust for QuPath on IMC data within QuPath itself, you'll be able to get good cell segmentation on your data sets. So that's one example.
So basically you are able to incorporate, because you can code into those softwares, right? So if there is anything available, and Stardust is the most popular or the one I've heard of, I assume that you can either develop, there are other models available that are open source that you can plug it in if you
could plug it in. So yes, you can, I mean, the challenge is it will require some knowledge of Groovy scripting, which is a little specialized, but Mark's very good at that. And so we write those scripts and then run them within QuPath. And then we also do other things. So QuPath has a pixel, sort of a tissue classifier built into it. And so we've done where we've collected annotations of glomeruli, tubules, interstitium, and trained a model to identify those regions within a IMC image. And then once we've done the segmentation, QuPath also lets you extract a bunch of spatial features from each cell in that image. And then what we do is we export all of those features, and then we've run XGBoost, which is a type of machine learning classifier, to predict the cause of a certain disease type based on just the intensities and the types of cells that were present in the image. And we're able to do a fairly good job, about 84% accuracy in predicting the cause of, in this case, it was transplant rejection, purely based off of the type and the location of immune cells that were present within the biopsies. So, and the reason we chose XGBoost, which is a machine learning classifier as opposed to deep learning, is that it's more explainable. So within XGBoost, you can actually get a list of the most important features that made it decide that this is one type versus the other. And so we could see the interesting, we could interrogate it a bit more than in deep learning, when it's sort of like, it's a little harder to make deep learning explainable.
Yeah, this is super important in both research and clinical, in the healthcare
in general,
space, in general, life sciences and healthcare space, the explainability is part of the validity of your data, I would say, because if you can explain it and show the logic based on, already phenotypes that are known, rather than end-to-end deep learning models, even if they work the same and have the same accuracy and they're just basically the same outcomes, the same results. And if you have it explained, it's a lot easier to accept by the scientific community. And that kind of, for me, is a stepping stone towards then accepting the end-to-end models that can also be super useful, right? But you have to know that it's repeatable within the end-to-end. It's rarely that you can explain it. But if you have something that does the same but has the steps explained, that has great value.
Yeah. And then another thing to mention also is, so as part of the open source, commitment to open source, I think one of the things that's been on my mind forever that I'm just working on implementing now is, let's say, a digital pathology wiki, where anyone can come and can contribute that, hey, I've just published this paper this last month. Here's the link to the GitHub code, and here's a link to the paper, right? Because I think the challenge that I have, that I'm sure I'm not alone in, is that there's like thousands of digital pathology tools that are all buried somewhere on GitHub. And, and it's sort of like, as either as a new user or even as an experienced user coming to this field, you could make the mistake of developing something for your master's project that already exists.
And see that already 5 people said that it failed. Or yeah, so this
Yeah.
is a big deal. I guess it's not digital pathology specific, but because we are working in this space, we notice it here. Like this concept of reinventing the wheel.
Wheel.
Like
Yes.
nobody wants to reinvent the wheel, but then once you started a project, nobody wants to abandon a project, and then you end up developing the same thing twice, three
Yeah.
times, or I don't know how many times.
Yeah.
And then you look up PubMed and it's like, oh, they already did that 2 years ago and it didn't work. No wonder mine didn't work. Or,
Yeah.
oh,
Yeah.
did
Yeah.
work.
Yeah.
Then, oh, mine is
Actually,
not such a fantastic discovery. Right. So
yes.
yeah, like a
Yes.
repository, that would be
Yeah.
fantastic.
And so I think, well, hopefully I will get it out by the time this podcast is out. But
Yes. So how far are you? How far
uh,
are you in the
it's
building? Show. I want to see
well,
it. Let's
yes.
talk about it.
It's, I mean, essentially, at a high level, I'm going to have like major categories, right? So you're going to have sort of pre-analysis, sort of QC, that kind of stuff. There's going to be a section on segmentation, section on classification, section on sort of like supervised and unsupervised methods, and then post-processing, right? Those will be the broad categories of like starting from the image going through to analysis. And then within those, what I do have is I've sort of collected a number of, let's say classical papers, let's say like Stardust, right? The first publication on Stardust would be a great one for the segmentation portion, and even stain separation, right? There's a number of different ways of doing stain separation to get from your H&E image to just H&E or H and DAB, and they all have their pros and cons, but whatever, let's just list them all and then let people decide as to, which one makes the most sense. And really, I mean, it's what's required on my end is to actually put that wiki together, which just requires a bit of fiddling with finding the right domain host that can let me install all of the right things. So it's not a big hurdle, and I keep talking about it, and I just need to do it. But like I said, I'll make it a goal that within the next month I'll get it up there so that we can put it on here, and it would be under the pathomics.io somewhere, pathomics.io / digital something, and that we can share.
I'm gonna definitely link to your website in the show notes, and if the thing is ready, then it's gonna have a separate entry in the show notes.
Perfect.
I would love that because it's perfect for pathologists or for non-computer scientists working with computer scientists, like tissue experts working with computer scientists, to point out to, hey, look what's already done, and let's not reinvent the wheel. Because I keep like repeating the slogan, stop reinventing the wheel, but I cannot point to what has already been done. It's on the computer scientist to research that. And obviously, when I do my literature research, okay, I find 5 papers and then I'm done. The most recent ones, let's assume this is the state of art, but I'm working on deepening my literature research. But basically, this is how it works, right?
For sure. Yeah, you look at a couple papers and you pick, but wouldn't it be great? And then actually, I mean, the fun thing is that with ChatGPT, right? We could ask ChatGPT, hey, give a list of all of these. So that's, I mean, we'd have to double check, right? Because half of the time it's making stuff up. But
check because it's generative AI. It's not a search engine. But yeah, you could basically have, let's fantasize. You could have your stuff already there and have ChatGPT work with the data that's already there and search or like answers based on this. So this is so cool.
And there is, I mean, there's a company out there that already exists called Hugging Face, and they're essentially a repository of all machine learning models. And in that instance, I'm just building a domain specific Hugging Face for digital pathology. So which should, so cool. Like as I said, isn't a big hurdle, but it's something that would be,
I'm going to be on the lookout for this because I recently opened a membership site, a paid membership site for digital pathology interested people, my digital pathology trailblazers. And what I'm doing there, actually starting by the time we publish this, it's going to be already up and running for a long time. But I started yesterday, every day reviewing like a quick paper review. And when this is out, then I'm definitely going to include this in our daily digital pathology digest. That's going to be a must.
Well, that's great. No, I think I've attended a few of your things leading up to the
Yes,
trailblazers.
yes.
It's great. Yeah.
So that's the newest thing. Okay, thank you so much for joining us today and explaining about Pathomics and about the commitment to open source. This is so cool because it doesn't mean that you cannot do business. You can do business and
You
still
can do business.
be
Yeah.
committed to open source and make it available to people at different level of financial resources, different level. I mean, there's nothing free in the world. You either pay with your time or with money. If you want to have stuff fast, then you pay with money. If you cannot pay with money, you do it yourself and you pay with time. So this looks to me like a tool for the whole spectrum of image analysis researchers, which is super cool to have something that's that accessible with the support of experts, and then with a cool platform that people can use in the making. Thanks so much for telling us. And you have a great day, Trevor.
Thank you so much. It's been really great talking to you, Al.
Thank you so much for staying till the end. The fact that you're still here means that you do resonate with the subject. So I'm going to leave a link to all the open source software tools that I know of available for tissue image analysis. And if you're a tissue image analysis scientist yourself and need to increase your pathology knowledge to be able to understand the images and the tissue in the images better, I have a course for you. It's called Pathology 101 for Tissue Image Analysis. And it is within the membership site that I recently launched, the Digital Pathology Club. So I would love to give you a free trial to the club so that you can take advantage of all the courses that are available at the moment and join a community of like-minded digital pathology trailblazers. So I'm going to leave the link below. Go ahead, click it, and give it a try. And I talk to you in the next episode.